Great. Well, welcome to Neurocare Academy. I'm Dr. Trevor Brown, and today we have with us Dr. Flavio Frohlich. How are you going, Flavio?
Hello, it's so good to see you, Trevor. How are you?
Yeah, I'm doing well, thank you. It's excellent to have you with us. Just to introduce you, Dr. Flavio Frohlich is the Professor of Psychiatry, Cell Biology and Physiology, and Biomedical Engineering at the Department of Psychiatry at the University of North Carolina and Director of the Carolina Center for Neurostimulation. So, wow, quite a title there. I'm sure you've done a lot of hard work in the past, Flavio, and I'm really looking forward to listening to you talk about neurostimulation today. So thank you very much for joining us.
It's a pleasure to be with you. This is very exciting.
Excellent. Very good. So, Flavio, look, we'll get straight into it. You've done, obviously from that title, we know you've done amazing work throughout your education. And I'd love to hear a little bit about your past, your background, where you were born, how you grew up, and where you did all your education. So maybe we'll start off with that.
Absolutely. Wonderful. So just real quick, before we get into it, just real quick, here are my disclosures. Nothing atypical. Ultimately, the translation of scientific discoveries into transformative healthcare interventions require commercialization, and I'm happy and proud to be part of that too. Also, in the form of disclosure, before we talk about myself, and somehow it is already about myself, I am not a medical doctor, so nothing I'm presenting or we're chatting about today should be misunderstood as medical advice, because I'm a researcher. Now, it's really exciting to get the chance to share with you all a little bit my own trajectory and where I'm coming from. But I want to just make sure we don't forget that a lot of the exciting science that I get to talk about to you today and also in other venues is really the hard work from my team, the Frohlich Lab at UNC at Chapel Hill. With that, if we go back in time, so I grew up in Switzerland, and my plans as a young adult were definitely not to study brain stimulation or do anything related to physiology or medicine. I went to ETH Zurich as an undergraduate student, which is a technical university, studying electrical engineering and management, thinking that maybe one day I'll be running a big car manufacturing company or something like that. And what happened there is that I really enjoyed the more abstract conceptual stuff in engineering, thinking about complex systems, thinking about how inputs and outputs relate to each other as function of internal states. And I think so often as an undergraduate, one gets pulled more or less randomly in a research group. And I ended up in a control engineering research group in electrical engineering, which essentially is the field of engineering where we typically think of complex processes, whether it's a chemical plant or the cruise control of a car or anything technical where we have input on the system through actuators, and we have sensor to measure the state, and we can connect the two to impose the desired behavior of the system through control design and through control engineering. Now, the specific application that I started working on still as an undergraduate research assistant was actually applying this engineering technology to the nervous system. So that's my first interaction with neuroscience. I knew very little about neuroscience. I taught myself how to model a single neuron and how it generates action potentials. And I applied the control engineering to it. And what you see here are my first two publications, where the idea was, as we're measuring the specific dynamics of a neuron and its electrical signaling, we can deliver targeted, you guessed it, stimulation to change neuronal activity. And actually in one of these two papers, and I put the feedback stamp on there because that's a theme throughout my career, to think about how to stimulate as a function of the state of the system we're measuring. Now, the ideas here presented in these papers are limited to an individual neuron, which is a much simpler case than what we're trying to work on together as a field today, which is controlling large-scale network dynamics across and within brain networks. But that's how I started out. So you start to see here a merging of fields and a new interest in neuroscience as an engineering student. And as part of my studies, I had the chance to spend a year at Imperial College in London, where I met this really fascinating person, Professor Igor Alexander. And at the time, he was a pioneer of neural networks, what today is called machine learning. But his research interest had shifted towards studying what he referred to as artificial consciousness. So consciousness in robots, consciousness of complex mathematical structures. And this is a quite unusual idea, but it was very exciting. But what ultimately happened to me as part of being involved in this research, that I got so curious about finally actually learning about the brain and how the brain works, and not just how engineers think about the brain. So that was my undergraduate education. And with that, the next step was to bring together these more mathematical interests with actual neurobiology. And I had the great opportunity to go to San Diego for my PhD, which was a big step for me. So I essentially emigrated and left Switzerland and moved to the other end of the world to do my PhD at the Salk Institute in UCSD. And what was really unique about the training I received there is that I had the chance to pursue and learn about more refined mathematical modeling strategies of neurons, and also learn how to do preclinical neurobiology experiments, because that's what I was longing for, to really understand how that theory maps onto reality. And here is some of my PhD work as we're talking about our own thinking, because I realized there is a theme here and I wanted to share this with you. So I was thinking about, again, network dynamics. So I made the step from individual cells to networks of brain cells in the context of epileptic seizures, where there's interesting transitions between different rhythmic states. Now, the mechanism I studied was actually extracellular ion concentration dynamics. And the details don't matter here, but you can see here on this slide or some of these plots, again, it's a feedback loop between neural activity, and in this case, another biological process, which is change in ion concentration dynamics, that then had, again, an impact on the neural activity. So it's everything in nature follows these closed loops, and you see the parallel to what we're trying to do with stimulation. So that's just the emerging theme of my training and my activities as a junior scientist. After that, I went to Yale for my postdoc, and the idea was there to learn how to test some of these ideas about networks and how they respond to perturbations in preclinical bench biology experiments. And I studied the effect of weak electric fields generated by the brain, so the kind of signals we routinely record with EEG. Now, the theme here was, again, feedback. And what we've done here, what I had done there was, first of all, understand if these weak electric fields, akin to what the brain generates itself, as an effect on neural activity, that you can see here very clearly in red compared to black, where the neural response or neural activity was present in that system, responded and trained to stimulation. So how does that fit in with feedback? Well, if the brain generates an electric field and at the same time the brain is susceptible to electric field, you have yet another feedback loop. So this is still continuing my journey, still continuing essentially an idea I first started to pursue as an undergraduate student in my research. And I built essentially a hybrid system, which was part biology, a brain network, and part engineering, to close that loop and study that feedback loop. And I found that this closed loop interaction enhances or enhances these neural rhythms. So this is a long story, but this is my training. I came from Switzerland from engineering. And as you realize, step by step, I've come closer to biology. And what I found most exciting about this is I was always able to bring in that different perspective, that control engineering, that thinking about complex dynamical systems, and integrate that with the things I've learned in terms of neurobiology. So those are perhaps the most salient influences that shaped my own development in terms of where we are today.
Excellent. That's amazing, Flavio. And I've been very lucky to interview people with a very, very wide range of skills, and it tends to be those people that get into neurostimulation as well, because you need such a wide range of skills. And with your mathematical background and your engineering background, it's a very concrete subject. And to now think about the human brain and consciousness, it's just such a broad aspect or broad view that you bring to the field, which is fantastic. So it's really exciting to hear what you've got to tell us about neurostimulation, the feedback loops, and the possibility of training the brain. So it's going to be wonderful to see what you have to share with us today.
Yeah, it's really interesting to think about how science evolves. I appreciate your observation about how people with different backgrounds come to neurostimulation. And my own experience has been that there is a lot of work that we need to do at the interface between different fields, right? And it's a lot about communication. It's a lot about language. And I think figuring out how to bring things together. So during my PhD, it was my attempt of bringing together computer simulations and experimental studies from a collaborating lab. And I realized how hard it is to build these bridges and just understand each other. But ultimately, I believe that's where the science gets really wonderful, when there's a shared language and we can work together. And the incentive system in science doesn't always support that, because in many systems there is incentives are assigned to individuals, and it's like winner take all approach. And I really find this is an obstacle to great science, right? So I very much believe in this infinite game mindset of where we're not in a competition for scarcity of resources, or which idea works better, or which simulation technique works better, but that all these different things fit together, and that's where the true synergies come from. So I've struggled with getting these different perspectives together and different labs to collaborate, mostly because I didn't myself realize how these systems work. So one thing that resulted from all that kind of at the meta level is that I wanted to realize my dream of building a research team which is fully integrated, where there are people who do computer simulations, people who do preclinical animal studies, people who do human studies, people who do clinical trials. And the vision was that everyone is always talking with each other and is willing to spend the extra time and energy to learn each other's language. And it's work in progress. So I've been at UNC now for a little bit more than 10 years, and maybe I can share with you a little bit how we've set things up and what we focus on.
Yeah, absolutely, Flavio. And with these days with COVID, our ability to actually mix and mingle in university has been obviously stopped a little bit, and with all the the distancing. So to gain the perspective of other people's fields is difficult nowadays, but it's so important in order to piece all this together. So looking forward to seeing your perspective and what you've done with your lab now.
Absolutely, that's wonderful. I appreciate the opportunity. So, where we are today, my office is in this building. Not that I've spent a lot of time in that office the last 18 months. Nevertheless, appreciate the facilities. So the way we're organized is, I really consider our team as having two main goals. One of them is we have this very ambitious, perhaps naive, but I believe very important vision that we can truly revolutionize psychiatry by developing new treatments based on the basic science, based on that integration between the preclinical and the clinical work. And then the second part is lifting up and enabling the next generation, right? So this is one of the main things we want to accomplish with our center, which is disseminate the technology, disseminate the knowledge, train the next generation. And I think that's very much aligned with the vision that you guys are all pursuing. So this is an exciting moment for me to talk to you about this. So if we talk about psychiatry, I believe we're in the middle of a big paradigm shift. Now, I want to fully recognize that psychiatric medication plays an important role in the care of numerous patients across the globe. So I just want to give that as an upfront statement. But in essence, where we're going in terms of the brain stimulation in psychiatry and other related fields such as neurology is we're really shifting from thinking about molecules to shifting about networks, right? And it's very interesting because the reality is there's a significant fraction of patients who don't respond the way we would hope to medication, and they can boil down to that incredible complexity at the molecular level, where perhaps very similar symptoms are caused by very different biological processes. So when we zoom out and start to study these network dysfunction, these dysfunctions at the network level, we might be close to kind of a common pathway where we're studying not the underlying genetic, molecular, biochemical processes, but we're actually studying the electric activity patterns that that generate the symptoms. So, and that's really fundamental, and it goes back obviously to the engineering way of thinking about systems, and it's the theoretical underpinning of at least of the clinical side of brain stimulation. And what's exciting about this is it brings in technology, right? So suddenly when we talk about those and other things like that, we really need to understand, physics, we need to understand electrical systems, and that then gives us that incredible power to come up with new stimulation paradigms, ultimately, I believe, individualized and adaptive for every single patient. So that's the view for the future. And it's based on all this, right? So we can routinely now for almost 100 years listen into the brain and record electric activity signatures in the form of EEG. Those recordings tell us something about the organization or disorganization of large-scale network activity patterns. And the step here, the next conceptual step, is to use brain stimulation to contribute, to support, to modulate how these rhythms are generated, to ultimately directly target the network activity signatures that cause symptoms and distress in these patients.
So look, that gives us a really good outline, Flavio, of your objective for really your life's research. And what have you started to do as far as your research is concerned? Have you started to really knuckle down on a particular topic at the moment?
Yeah, absolutely. I'm happy to share. So we've been going many different directions, but I have a few slides to give you an idea of where we stand today in terms of the overall goal for us specifically is to figure out how to best interact with these brain rhythms, how to in a targeted way restore them and demonstrate their causal role in disease process and thereby essentially develop new treatments. Here's an example of where we are today. So this is from invasive stimulation, but I feel it's a great example to conceptualize the type of work we do. So this is from patients who have electrodes implanted for clinical reasons. These are patients with medication refractory epilepsy. But what we've done here is we're giving them cognitive tasks to measure their brain activity, very similar to a standard cognitive neuroscience approach, with the important difference that we have electrodes in the brain and we kind of have a finer resolution of what's actually going on in the electric activity signatures of these networks. And what we found is, what has been known for a while, that in this case, working memory function is supported or at least correlates with synchronization as large-scale cortical networks. What we then did, which is truly novel, is we stimulated through these same electrodes right in the brain in different ways, synchronized, matched to the peak frequency of that activity signature we detected, or out of synchrony, out of phase in several different configurations. And when you do that, you have two important questions. The one to the left is, does the stimulation actually change the brain activity the way you anticipate? So we refer to this as target engagement, right? We identified that a cylinder synchronization that is correlated with cognitive function. Now, can we actually go and in a targeted way change that synchronization, which is what you see here to the left relative to a placebo or sham stimulation. And the other question then is, does changing these brain network activity patterns actually have an impact on cognitive function? In this case here shown accuracy and then also reaction time as working memory tasks. So those are the two key things to think about. right? One, can I modulate the brain the way I want? Two, once I succeed in that, are there actually tangible benefits at the behavioral level, whether it's improvement of cognition or whether it's decrease in symptoms? So how do we go about this, right? How do we know how to stimulate? Well, first of all, thankfully, very few people need invasive electrodes. So the beauty is, and I've already talked a little bit about EEG, the beauty is we can do this all non-invasively, right? So here comes tACS, or tACS, as some people say, transcranial alternating current stimulation. Derived from that insight that brain rhythms respond to meet periodic perturbations, we and others have developed tACS, which is essentially the application of a weak low amplitude rhythmic stimulation waveform to the scalp. And the beauty of tACS is, in contrast to other brain stimulation modalities, is that it's a small battery powered device because it's such low amplitude stimulation. And we have a good mechanism of action. And that's what I would like to share with you, because having a therapeutic gain is important, but if we understand what the stimulation does, how it works, that gives us a chance to make it better and to individualize it, to address a significant fraction of people in whatever treatment modality you look at that don't respond. So it's understanding the mechanism of action to enable what I call rational design, which is what we've been focusing over the years, and I'll have some results to share with you.
Thanks, Flavio. Really looking forward to seeing the results of that. And tACS is such a fascinating modulation tool that we've got, because it's so flexible, isn't it? You've got different frequencies that you can use to modulate the brain. You can link them to different frequencies that are innate in the human brain, such as the 10 hertz alpha frequency, and I'm sure you'll be talking about those different modulation frequencies that you've been using. But before we get it stuck into the real science of it, Flavio, I've got a little bit of a curveball question for you, just to get to know you a little bit and break things up midway through the interview. I was going to ask you if you could invite 3 people to dinner for a dinner party, who would they be, and why? Who do you find interesting and you think might get along together?
That's a wonderful question. I could think of many people, but perhaps in this context, I would like to reunite the people who have in many ways have started our field. So Luigi Galvani gets credited for that really important discovery that electricity is the currency by which the nervous system acts on the body and controls the body. Now, in reality, it's actually his wife Galeazzi who made that true discovery. So inviting the couple and perhaps sharing how much or how little progress we've made in terms of recognizing the true contribution by the scientists who we've unfortunately have marginalized over time would be wonderful to see that play out. And then a third person would be Alessandro Volta, who was kind of on the verge of making the same discovery, but I think was a little bit behind and ended up in a really major feud with Galvani. And I think reuniting them would be great, because ultimately, as it turns out, they were both right in their own way. They just approach it from 2 different perspectives. So Galeazzi and Galvani came more from the anatomy physiology side, versus Volta came more from the physics, which was still an emerging discipline at that point side. So getting them together and talking about, how in the abundance mindset, credit should be shared, and creating a little bit more fairness ultimately in the history books of who discovered what, and bring them together and demonstrate that if we work together, we can make much bigger steps towards ultimately what today is improving humanity, namely brain stimulation. I think that would be a really exciting and spirited dinner conversation that I would feel incredibly privileged to be part of.
Okay, so let's get on a bit more towards brain stimulation and psychiatry and treating mental health, and let's have a look at what you've got for us.
Absolutely, my pleasure. So in many ways, here is where it all started. So these are results from a computer simulation where we went after a question we and others have been looking at so many times since then, which is, how does the brain respond to these weak perturbations? And what we've done here in a computer simulations, we've tried different stimulation frequencies and different stimulation amplitudes, and that's the organization of these little plots, and every little plot is color-coded, is essentially the effective stimulation. You see these rhythmic patterns reflecting overall network activity structure. When you look at that, you see this inverted triangle centered at 3 Hertz, which is the frequency of the network itself, its endogenous frequency. And essentially, these computer simulations showed for the first time that when we think about weak perturbations such as tACS, it is important to tune the stimulation to the endogenous activity pattern. What's also interesting about this, if you shift your gaze to the right and look at those dark blue parts, you can actually have a counterintuitive effect of stimulation where the stimulation reduces the oscillation instead of strengthening it. And we found this in computer simulations almost 10 years ago. And while this was the first time described for tACS, the general principle has actually been known for a very long time. It's called the Arnold tongue. And it's been first described in the context of entrainment or locking of a system to periodic input in mechanical clocks and metronomes, devices like that. But it's really foundational because it tells us how to stimulate when we want to target a specific frequency. And it also helps us understand something that the field has grappled with quite a bit by now, which is why can such a weak perturbation, the resulting electric field is so weak, the change in the membrane voltage just from stimulation is so low, how can it have an effect? Well, physics has the answer to this. In the case of perfect tuning, an infinitesimally small perturbation can adjust the phase of an oscillation or entrain it. So it was really quite spectacular to find that same principle, you know, when looking at the output of these complex mathematical models requiring large-scale parallel computing infrastructure at the time, to find this beautiful theoretical principle embedded in how tACS works. And that is still just a theory, right? So going back to this theme of bringing together different fields, what I'm so excited to share with you is that very recently, actually published this year, with the support of the NIH Brain Initiative, we had the chance to test this in an intact brain. And without going into details, if you look at this top left plot, you again see this inverted triangle, which indicates that indeed, if we apply a low amplitude electric stimulation, the amount of entrainment or synchronization depends on the tuning of the frequency. Now, there are more details to this that I'm excited about. So if you compare left to right column, you see that to the right, there's much less of an effect. So these are actually the regular pyramidal cells. So what we found here is that it's really the inhibitory interneurons that drive that synchronization endogenously, and also the ones more responsive to the stimulation. Final point about this, you don't see that structure in the purple 2 plots at the bottom, but these are overall firing rates. So that goes back to a confusion the field has been having the last few years, because people were thinking if we stimulate, we should see a change in firing rate. But no, we don't, because for weak stimulation, all we need to do is we need to shift around the action potentials, and thereby we can augment synchronization, rhythmic structure, and thereby we can facilitate the information flow and improve cognitive function or relieve symptoms as a function of application. So it was a very satisfying moment to get the chance to, I remember when these data first came in, to see that this theoretical prediction of mechanism of action that we had published almost 10 years earlier, was there as expected, with this nuanced twist of the cell type specificity in these preclinical animal models. So this is what a lot of what we're doing today is based on, and that there, I think at this point, there are many labs around the globe who pursue tACS for exciting applications. And I think one of the main messages that has resonated by now is that weak perturbations can have a profound effect as long as you smartly tune them to match the specific intrinsic network dynamics that you want to target.
It's so exciting because, I mean, obviously throughout mental health work throughout the decades, there have been a lot of invasive techniques. I mean, you know, you have the adverse side effects of ECT. ECT has done an amazing job in helping a lot of people in depression and other disorders. But then you have the very invasive techniques like deep brain stimulation and even non-invasive techniques like TMS, which I know you work on. It's sending 3 to 3 Tesla of electromagnetic field into the brain, whereas here we've got a very, very weak signal, and through entrainment can actually have a large effect. And that's really exciting because it's so non-invasive and it's entrainment.
Absolutely. And, I think it's really important, as you just did, to acknowledge that there's an entire ecosystem of approaches, and they all make sense. They all have different levels of science behind them, and a lot of them, such as ECT, are clinically incredibly effective based on what we know from the literature. So I think all these different modalities can coexist. And what I want to make sure is we never think about trading one versus the other or competing one versus the other, because they all have unique advantages and disadvantages after use cases. And there's still so many patients who don't get any brain stimulation treatment, right? Again, the abundance mindset, there's more than enough for all of us to study, because ultimately we all have the same goal. And if patients get better, it doesn't matter to me whether it's a thing that I study or someone else studies. So, but I'm glad you bring up TMS, because I think it's a really interesting comparison to think about this. So TMS is about 100 times stronger than tDCS or tACS or in general transcranial current stimulation. So that already tells us that the two stimulation modalities can do very different things. And it's not one is better than the other, but it's really because while it's still delivering electricity, because TMS uses magnetic fields, we have the ability for spatial targeting. So I want to share with you the results of one recent study, which has become quite controversial, but I find exciting because one, it demonstrates the synergistic coexistence of electric and magnetic stimulation. And two, it goes back to what you just mentioned, that really important point that weak perturbations can be very effective. So, as you know, about 20 years ago, there were these initial papers suggesting that a constant current applied, a very low amplitude constant current applied to the scalpo-motor cortex could change motor cortex excitability. So just to put us quickly back in that moment of time, this was in many ways an old idea, because people have talked about brain stimulation through, for example, natural means for multiple millennia, but in the same way it was also a birth of a new generation, in this case of tDCS research. So stimulation of motor cortex and application of then TMS to target a specific motor circuit, and then measuring the evoked output in the muscle in the hand, right? Classical experiment. Everyone familiar with who works in that field. But what's exciting about this, besides that it brings these two methods together, is we went after replicating that not that long ago. And the reason we've done this was basic curiosity, essentially. We focused so much on the entrainment and the synchronization. We were just curious, what does actually a constant current do in our hand? And it was also a very humbling lesson for me, because sometimes as a scientist, you'll get locked into the one thing you're doing and you think that's the answer to everything, but it's really not. So here are the tDCS results. Essentially what you see here to the bottom left, the results were incredibly crisp and clear that as a function of current direction, the way for 20 years paper have suggested it would happen, also in our hands in a crossover, double-blind, placebo-controlled study, we had a very clear modulation of motor cortex excitability. And not only that, we also had a corresponding modulation of evoked potentials in the brain measured with EEG that correlated with that change in motor output in the muscle. So there are many studies like that. I'm just sharing the one that we've done because, while I often talk about tACS and brain rhythms, I feel it's a very important example to illustrate that the value of bringing together different approaches. And also I find this is perhaps one of the clearest answers we have so far about what exactly a weak electric field does, and that indeed has these effects. confirming many of the, and replicating many of the previous studies, but also extending it because showing a clear brain correlates. And the funny thing about this is, like originally was, people who study tACS, they think tDCS is not good enough, but it's really not true. Again, it does two completely different things. This is a plasticity effect that's very different from what we're doing with tACS. And the other interesting thing about this is, the effects are so small, they're not really clear. And then once you get a strong and clear effect, then people go like, oh, but how can you get such a strong, clear effect? And then you talk about, how to do well-controlled studies and all the extra steps we've undertaken here, which were quite exceptional. And a lot of credit goes to Sang-Dian, my former postdoc, who's now faculty himself in Korea, to get to that point. But anyways, take a home message. There is no competition. We can bring the different modalities together. And yes, absolutely yes, weak electric fields change brain excitability in predictable, albeit complex ways.
All right, so that's fantastic, Flavio. Let's knuckle down into the sort of the nuts and bolts of your current research. I'd love to hear about what your lab's getting into at the moment.
Very excited to share. So the overall principle that we're employing is essentially thinking about brain stimulation as a platform technology. And what that means is that I believe there will be many different applications, whether it's in psychiatry or neurology or related fields, where brain stimulation can be incredibly beneficial. But to be really clear, I don't believe everyone gets the same stimulation and it's one thing that works for everything. I think that would be a very naive and inappropriate assumption, but rather what I believe is we can leverage neuroscience and some of these more basic science approaches we've briefly discussed to do rational design. So what do I mean with that? Well, the 3 steps, and I have it here illustrated from a clinical trial in chronic pain that we've recently done is, first of all, you need to know what your target is. Just to stimulate because you want to stimulate is probably not good enough. What is the specific activity pattern? And this is important because, as we've discussed, we understand the mechanism of action of tACS. It's entrainment by the Arnold tone. And by definition, that means we need to know what the endogenous rhythms are, not only where they occur in the brain, what frequency, but also perhaps how they're different from person to person. So we need to find an activity pattern that correlates with your construct of interest. In this case, it was chronic low back pain. So the idea was that sensory motor circuits, as others have suggested, for chronic pain could be hyperexcitable, which would correspond to a lack of inhibitory top-down control, which is often the alpha oscillation that you have previously mentioned. So we looked and we found in these patients a negative correlation between symptom severity and the strength of alpha oscillation. In other words, the more symptoms a person had, the more decreased those alpha oscillations, or perhaps some people refer to them as mu rhythms in the alpha frequency were. So that gives us a target. And for many indications, we can go to the EEG literature and find those targets when people describe this and this patient sample, perhaps compared to a control group, and is demonstrating or reporting changes at the EEG level of these brain rhythms. Now, then the next step is leveraging that insight and the mechanism of action to design a stimulation paradigm that goes after that specific deficit and demonstrate that yes, indeed, you have that effect on brain activity. So what I'm showing you here is in bright red in that topographic map in the middle is that essentially we're able, in comparison to placebo stimulation, to strengthen selectively that rhythmic activity pattern in that part of the brain. Finally, perhaps the most tricky, but also the most important one is ultimately you want to be able to demonstrate that the changes in brain activity that you cause by stimulation correlate with the change in symptom. And that gives you 2 things, right? That gives you, you're going from a correlation to causation in terms of understanding mechanisms of disease states or cognitive processes or consciousness, whatever you're interested in, on the one hand side. And the other hand side, what it also gives you is an actual intervention, and for which you have the mechanism of action that you can personalize and individualize. So To my knowledge, we were the first ones to do double-blind placebo-controlled studies of tACS in psychiatry, and I want to share some of these results with you to give you an idea what that looks like. So here's a study in patients who have medication refractory auditory hallucinations in schizophrenia. We had a similar hypothesis about a lack of top-down inhibitory control, this time in another brain area, in the more language, verbal areas in this kind of frontal temporal network. So what we've done in a placebo-controlled parallel group design is we have applied tACS to target these networks, and what you see here in this plot is essentially the alpha peak and its spatial distribution and how tACS strengthened that alpha oscillation. And that study, I believe, is key because, to my knowledge, it's the first one that shows multiple days of stimulation in a patient population within a double-blind placebo-controlled study, that you can indeed augment this targeted brain activity, and also that the effects persisted, at least at the numerical level, for weeks thereafter. So this was after a 5-day paradigm. So with that basic insight that we can alter, perhaps even outlasting way, these network activity signatures, we then went and asked, how about other aspects of auditory processing that we know is altered in schizophrenia? And one of them is the response to a 40 hertz click train. And what we found here, what you see here in red, is that actually at stimulation, while we didn't target the 40 hertz, we targeted more that overall organizational top-down inhibitory rhythm. Actually restored some of that auditory processing that is impaired in schizophrenia. So that demonstrates that if we target one specific network activity signature, we should also think about the other processes, because all these different rhythms as a function of context, here in this case, in response to presentation of a computerized auditory stimulus, they all relate to each other because they're ultimately all generated by the same brain network. And I think that's something we can focus on much more in the future, instead of just targeting one brain rhythm and be locked in one thing, to take a much more holistic neurophysiological, but also behavioral assessment of how stimulation reorganizes these brain networks. Now, that is interesting neurophysiology, but the key here is, and I found this quite remarkable, that when we looked at how much that response to auditory input improved on the x-axis of these plots, that actually negatively correlated with symptom severity of auditory hallucinations. So the more we manage to restore the basic neurophysiological property of how we respond to a click train, essentially helped us understand to what degree patients would improve symptoms-wise. And on the y-axis here, that auditory hallucination rating scale is a very holistic measure. So it's not just the basic auditory percept aspects, but it also includes aspects such as controllability of hallucinations and the distress that they're causing. So early studies that will need to be replicated, and we're currently analyzing a data set from a follow-up study. But the bottom line is this got us really excited that this approach could indeed play out. So where do we go from here besides replicating, working on a study to replicate these effects? There are other things that really are unique about this technology. And one of them, it's a risk benefit profile, because it's incredibly safe. So here we've done in a single case of a patient with medication refractory auditory hallucinations within a feasibility study of weekly stimulation. So this is 20 weeks of weekly stimulation. So that's a completely different paradigm that we're now working on studying at home. And what happened here really demonstrates how important it is for different fields to come together. If you look over 20 weeks, just at that overall rating, holistic assessment of auditory hallucinations, there was a little bit of a change, but not a very pronounced change. Yet the participant reported subjective feeling so much better. And what we then found out through qualitative research methods based on comments they had shared with us is that something had changed about the controllability of oscillations. So the patient reported about halfway through that they feel they have some control over the occurrence of these auditory hallucinations. And that was really interesting. So we could then go back to that psychological clinician administered assessment and pull out the items that relate to that controllability. And you can imagine how important that is for quality of life. And that's what's shown to the right. And there's indeed quite a significant and impressive change. So the reason why I bring this up is because that's kind of the other end from the basic neurophysiologist shared with you, right? So there are many different perspectives. This is really at the highest order, almost, at the meta level, how a patient conceptualizes and experiences distressing, very complicated, severe mental health symptoms, and what the stimulation perhaps could do, which lines up with all we know about alpha oscillations in general, about cognitive control and top-down control. So there are many things I like about this case report. One of them was a truly collaborative nature with the participant, which I think is something we all as researchers can really focus on. We can learn a lot from the patients in our studies beyond standardized assessments, one. And two, you can see here that all the electrical engineering in the world doesn't help. I mean, yeah, you need to build the box that works safely and all that, but the psychology and the psychiatric perspective here is equally important. And I think, efforts like yours that we're now part of with this conversation are so important to bring people together and again develop that shared language. So I thought it was a exciting example to share. I want to shift to depression because obviously it affects a tremendous fraction of people across the globe, and clearly COVID has made it much worse. So some of the latest data out of the US shows that about 50% of all households in the US have someone who is affected by depression or anxiety. And it also gives me a chance to go back to TMS. So you see here a TMS setup, and as you know, TMS has been in many places cleared and approved as a treatment for depression. So TMS is in that way ahead of tACS. It's an exciting stimulation modality that's bringing more and more people relief. But the question here is, what is actually the mechanism of action? And when we know the mechanism of action, can we improve and individualize the treatment? And furthermore, because TMS is also a rhythmic stimulation modality, maybe there are some shared underlying principles that work across the spectrum of different brain stimulation modalities, that most of them, not all of them, most of them are rhythmic in structure, and thereby perhaps actually target brain rhythms. And I have here an image from a paper from Andrew Leuchter and colleagues who was one of the pioneers suggesting, well, the 10 hertz at which we typically do TMS is really right in the middle of that alpha frequency. So let's start thinking about what the network level target engagement and mechanisms of TMS for the treatment of depression actually are. And that inspired us to think about tACS. But first of all, what exactly are these alto network activity patterns? And there's no single clear biomarker of depression in any way, and definitely not in terms of EEG, but the alpha oscillation has been going in and out of favor as one potential mechanism. And what is really exciting, the last few years, I've seen a resurgence in really high quality research demonstrating, one, the role of alpha oscillations and alteration depression, and two, show a relationship to how we stimulate with TMS and what kind of clinical benefits we can derive. But we made a step back and just wanted to make sure that this basic idea of an asymmetry between left and right frontal alpha oscillations, which isn't per se validated as a depression marker, but there's quite some literature matching it onto more trait level features in terms of approach motivation or behavioral inhibition as a function of hemisphere, whether we could find the same thing in healthy controls of patients. And long story short, of all these data points on this slide, we're able to replicate that finding that inhibition of left frontal cortex maps onto a reduced approach motivation in the behavioral approach system, and inactivation or inhibition of right frontal cortex essentially results in a behavioral disinhibition or a reduction in behavioral inhibition. So that's really exciting, because while the simple storylines of measuring alpha oscillation and you have a biomarker for depression hasn't panned out, underneath there, as we start to unpack it, there's a lot of really exciting neurophysiology that I think will help us identify targets for stimulation. In fact, in additional twists to the story, we found that this alpha symmetry that has been quite hotly debated the last few years, actually partially, the inter-individual differences thereof can be explained by testosterone levels. And that again speaks towards bringing together different people. Here we had wonderful collaborators in the who studied the role of sex hormones in mood disorders and related processes. So, and there was also a sex-specific effect where we found that relationship only in women. So as we start to unpack and as we start to add in other parts of biology that I wouldn't have thought about 10 years ago when I was still thinking of the brain as a strict electrical system, right? Without that, we would miss all this. And, what I'm also showing you here, this is work by a great postdoc in the group, Dr. Riddle, that we can actually localize where these signals come from and think about how and where to target them with TMS. Now, in this case, going back to tACS, right? What about 10 hertz tACS? And so we've done now 2 studies I want to briefly show you in people with depression or in a major depressive episode, where we try to target and rebalance frontal alpha oscillations. So you see here the stimulation waveform and the resulting predicted electric field strength. So this was an RCT, I believe the first RCT of tACS and depression. And it was a pilot study where it is a double-blind placebo-controlled study. And what you see here to the left is that the vast majority of people responded or had at least a significant symptom reduction when they received 10 hertz tACS versus while there was a placebo response in the placebo group, it was much more varied, and on exploratory analysis, there was a significant difference in terms of fraction of people responding. So that is a behavioral effect of stimulation, and I think we can put some faith in it because it's a result of an RCT, and we were able to demonstrate that the blind was maintained. But really, after all the neuroscience I shared with you, right, like what is actually going on in the brain? And what we found is, as predicted by this idea of stimulating left and right in phase, that you would rebalance alpha oscillation, we found a reduction in left frontal alpha oscillation was specific to tACS, demonstrating, and this was the primary outcome of the study actually in terms of neurophysiology, that the stimulation had the desired effect. So that was a relatively small study, but it made us incredibly hopeful that we were on the way towards understanding how we could use tACS to treat depression. It's still early days, and any study, even carefully done studies, need to be replicated. And we've done one step in that direction. This was not a treatment study, but we just people that come in for a single session. And we've done the same stimulation, both in actually patients and healthy age and sex matched controls. And what we found is what you can see here on the left-hand side, and again, this work by Dr. Riddle in our group, we found a decrease in left frontal alpha oscillations with stimulation, thereby, I believe for the first time, actually replicating with a larger sample the effect on these pathologically altered brain network dynamics by tACS.
Thanks, Flavio. Look, what I find amazing with your work is, well, to begin with, a few things, but to begin with that case study that you showed us and trying to disentangle the auditory hallucination decrease in symptoms with the decrease in alpha, and just after 9 or 10 sessions there, the switch in quality of life and being able to control those auditory hallucinations. And that's really hard to really, when we have these controlled experiments, say, okay, we're controlling for auditory oscillations with alpha, and what effect does that have in such a complex environment like the brain? It's so interconnected, it's really hard to predict the value of a treatment like tACS. We can say that we're manipulating or entraining the 10 hertz alpha rhythm, and we're going to get a decrease in auditory hallucinations. But the global effect on quality of life can be so much greater than that as well, and it's sometimes so hard to measure. But look, that's fantastic. Another point there is your final point in that the biomarkers for depression, it didn't really come through, did it? The left decrease in activation and the right higher activation in depression as a biomarker, but as a predictor of response and as a target for training and a target for intervention, it is quite something that we can use to personalize intervention. And that's been happening in not only TMS, but and now can start to develop in the tACS and tDCS as well. So yeah, brilliant. Sorry to just sort of paraphrase what you've just said, but I hope I've understood. And not everybody's a neuroscientist that looks at these interviews, and I try to sort of help everybody by paraphrasing a little bit of that as well at the same time. But so that's what you've been doing at the moment, Flavio. And just to finish off with, I'd love to hear a little bit about what you've got in the future. What's your future research oriented towards?
Absolutely, yeah. And let me just echo, I loved your summary, but let me also remind everyone that these are initial studies, right? Most of them were the first of its kind in any indication. It looks very exciting, and I think we've overcome those concerns about what is the effect of weak stimulation through the discovery of the mechanism and validation of the mechanism. But it's still early days, so a lot more research is needed. But I think everything I am seeing today makes me incredibly optimistic that we're on the right path. So what are the next steps after this? So where do we go from here? I believe one of the key things we can do now is, because we have this understanding of the mechanism of action, is to really talk about personalizing the treatment. So a lot of tACS work, including ours in the past, used an incredibly simple waveform that matched brain network dynamics, perhaps just in one feature, namely its frequency. But no EEG signal I've ever recorded looked like a sine wave, quite the opposite. Typically, there are lots of different rhythmic activity structures stacked on top of each other. So one way how we can personalize, again, by using this principle that the stimulation match the network target, is to add additional complexity or features to the stimulation waveform. So I'm just showing you here one example of where the stimulation waveform is including two different frequencies. So one nested into the other, what some people refer to as phase amplitude coupling. But what's really unique about this, and what we've done here, is we can actually, based on an EEG, find those specific frequencies and how they interact person by person, and adjust the stimulation accordingly. And while I've never done direct comparisons with and without that individualization, I believe if you have a lot of studies that don't show you an effect, I think it might be, based on all the mechanistic work we have now, it might be a good moment to reflect on, to what extent the individualization for better target engagement could improve the outcomes in your study. So that's one way how to personalize it, essentially thinking about time or the temporal dynamics or structure of brain activity that you target and thus of your waveform. The other one is space. While delivering electricity to the scalp is kind of by definition a more diffuse approach in terms of where the electric field gets delivered, but still with computer simulation of the underlying physics, we can come up with good predictions where the electric field goes, as shown here to the right. And we can use that to think about new and different electrode montages or configurations, perhaps with multiple channels, where to put the electrodes to engage specific targets. So if you take these two approaches together, time and space, and think about individualization in a kind of a personalized medicine approach, I believe that's one of the most exciting and promising next steps we can undertake together as a field. Now, the next step after that is to automate that. And we're kind of back at the beginning of our conversation, Trevor, because now we're back at this feedback idea, right? So I started out my career 20 years ago thinking about and demonstrating computer simulations, how to turn on and off a single neuron by interacting with its rhythmic firing. And where we are today is that we can close that loop at the network level. And this is a study we've published a few years ago by a postdoc at the time in the group, now faculty, Dr. Lustenberger. And what we've done here is we detected sleep spindles in real time and applied stimulation through a control algorithm whenever we detected a sleep spindle. And when we compared that to placebo stimulation, we found two things. One, we increased brain function by strengthening the activity in that specific frequency band of spindles, showing successful target engagement. And two, when we looked at the performance of a task that has been correlated with sleep spindles, in this case, motor memory consolidation, procedural memory consolidation, we found overall a clear effect of strengthening memory consolidation in comparison to a sham or placebo control. So that is the next level, to take all our mechanistic insights that we have about how tACS works, to personalize, individualize, and ultimately make it adaptive. And I have fewer thoughts summarized here to go even beyond that. So what is coming next? Kind of my personal predictions and hopes and wishes for the field, if you will. One, the general principle I strongly believe is the more we match our stimulation in complexity or in structure to what the brain is trying to do, the more successful we'll be, the larger the effects, the better ultimately the treatment outcomes when we're at that level with the studies we're doing. Closing the loop in many different ways, that is key to it. Like, we are individuals who sense the environment and adjust internal processes to interact in that environment. Everything is closed loop, everything is regulated. As I've shown more than 10 years ago, weak electric fields generated by the brain are not just nice for us that we can record EEG, but no, they feed back and shape activity. There's also a more procedural thought that I want to leave you all with is, it is comparably easy to do a pilot study of this and that in few patients and be excited about the progress. But the reality is the most profound medical intervention that we have today that works across pretty much everything, to my knowledge, is the placebo effect. And the placebo effect is real. There is a clear neuroscience basis. And we need to do double-blind placebo-controlled studies following rigorous gold standards, such that we get accepted in the field, that we can make true progress ultimately towards regulatory clearances, reimbursement by payers, and ultimately to revolutionize psychiatry. We need to bring these devices to people's homes, and the only way to get there is through an expansion of the rigorous science from the basic mechanism all the way to placebo-controlled studies. If I have any impact on the future, and I don't know if I have, but if I did, what I would wish for us is a rapid growth of placebo-controlled, high-quality studies, leveraging some of these principles that I've shared with you today. And we've recently published two opinion papers or little reviews. One of them focuses on, what do we need to do to take the closed loop into the clinic? What are the missing steps? Where is the opportunity? And the other one was actually a pretty much play-by-play instruction all the way down to the nitty-gritty details, how to perform a gold standard clinical trial of tACS. And I hope, going back to kind of the overall personal arch here, Trevor, is that with everything that our group has developed, where we were, standing on the shoulders of many giants in the field, that collaboratively we can all move forward and inspire the next generation to pursue some of these approaches, build on it. And certainly there's, as always in science, there's lots of things to improve and take this to the next level. And I believe that is how together we will change the world by revolutionizing psychiatry.
Great. Well, that's a great way to finish off, Flavio. A great summary of where the field's at and what needs to happen over the next 10 years. I mean, obviously, studies need to be replicated, then they need to be replicated by individual teams, and then there needs to be meta-analyses and clinical studies where things things do take time. And a lot of the studies that you've done with small sample sizes and pilot studies that you've shown us are the first step in a long journey to bringing this to the patient. So over the next 10 years, 15 years, Flavio, have you got an opinion about where the field is heading? What could you say to new psychiatrists, psychologists in the field who are considering neurostimulation, adding that to the practice, and how can you finish off? I know you've got sorry, this is a multi-level question, but also over the next decade, how do you see the field moving as far as the complex systems? I know you have this theory or a view or vision of the brain working like music, and would you like to sort of expand on your idea and tell us a little bit about that?
Absolutely. Yeah. So I think the main message is the science looks very, very promising, but it does take time, and it's sometimes hard. It's sometimes hard for us to be patient, and it's even harder to be patient if you're a patient. But I think the good news are that there are in many places, there are cleared and approved non-invasive brain stimulation approaches, such as TMS and also others for the treatment of various psychiatric and also neurological disorders. So I am not a clinician, but I think from a patient advocate perspective, thinking about providers who have that skill set or know how to refer to such treatments, it's often giving a lot of hope, because these modalities work very different from, for example, how medications work. So I believe there are two things. One of them, some of the stuff is already here today, and studies show is quite helpful to a significant subset of people who don't respond to pharmacological treatments. The other side to this is we can do much better, but we need to do more research. And we talked about all that electrical engineering and the complex systems, and I think that's one perspective I'm very excited about. But perhaps we can zoom out even more and think about what rhythm and synchronization really means in biology and for us humans in general. And that takes us to music. So what you see here is what I believe one of the very first EEG studies, where we've recorded from 4 brains simultaneously, a technique sometimes referred to as hyperscanning. These 4 people engage in a shared collaborative activity that is guided by a shared understanding of time or of rhythm. It's music. It's a classical quartet playing together. And of course, talking about feedback, when you play like that, you're listening and you're adjusting your own actions to find onto that shared clock. Now, in the case of a quartet, it's distributed organization. There is no conductor. In a larger orchestra, there's a conductor who might be a little bit like tACS, providing that overall input to facilitate the temporal coordination. So while not all of us are classically trained musicians, but many of us, most of us enjoy music. And we enjoy music because I believe, and that's a speculation, music is organized structure of time, which reflects very much what brain activity is. So I think the overall speculative thought I have here is that ultimately thinking about rhythms and manipulation of rhythm, whether it's through neurofeedback, whether it's through TMS, whether it's through tACS, whether it's through something new we haven't even developed yet, or whether it's through music education and training, whatever it is, ultimately I envision there will be one fundamental model which focuses on the fact that everything in nature ultimately is rhythmic. And it goes all the way back to the day and night cycles. It goes all the way back to how we periodically sample the environment, from breathing to moving our eyes to whatever it is. Rhythm is inherent to any complex organized system under most circumstances. And we have numerous abilities today, and we'll have more tomorrow, to leverage that fundamental principle of life for therapeutic benefits to make this a better world for everyone.
That's a brilliant way to sum things up and giving us a sort of a worldview that you have there, Flavio. That makes a lot of sense, and it leaves us with very optimistic about the way to treat mental health and performance in the future. I mean, the feedback loops that we're making use of, the ability for small perturbations in these in the EEG can possibly have an effect, and you're really with that oversight of music giving us really the closed loop perspective, which is really exciting. Thank you very much for your time, Flavio. We could sit here and talk all day if I had my way, but it's been a pleasure to have you.
It's been so wonderful. Thank you so much. What a unique thing you're doing, and I'm honored to be a part of it. This was wonderful. Thank you, Trevor.
Great. Have a great day. Thanks, Flavio.
Take care. Bye-bye.